Hi everyone, in this biochemistry, we would like to discuss on glycolysis, Krebs cycle and also electron transport chain. First step is glycolysis, which is breakdown of glucose. The first phase, glucose will be breakdown into fructose 1,6-biphosphate and the second phase into 2-pyruvate acid. There will be anaerobic glycolysis, aerobic oxidation, and aerobic alcoholic fermentation. Glucose undergo phosphorylation, addition of phosphate group utilize 1 ATP to form glucose 6-phosphate. That being converted into fructose 6-phosphate by isomerization, and for phosphorylation occur utilize 1 ATP to form fructose 1,6-biphosphate. Fructose 1,6-biphosphate will undergo cleavage to yield 2 3-carbon molecule, and one of it is gracilidehydrogenase phosphate. This molecule undergo phosphorylation, donate 1 phosphate group to produce 1 ATP, undergo dehydration, which forming phosphenol pyruvate that convert into pyruvate. In absence of oxygen, NADH will be oxidized and pyruvate will be reduced to lactate. Presence of oxygen will be proceed to citric acid cycle and then undergo oxidative phosphorylation to form carbon dioxide and water. Usually in use, NADH will be oxidized while pyruvate acid will lose carbon to form carbon dioxide and also ethanol. Next is Krebs cycle or citric acid cycle. Pyruvate from glycolysis will be oxidized to acetyl-CoA by pyruvate dehydrogenase complex that generate carbon dioxide and NADH, and then will be combined to oxaloacetate by citrate synthase to form citrate. Citrate will be isomerized to isocitrate by isonitrate, while isocitrate will be oxidized to alpha-ketoglutarate by isocitrate dehydrogenase and release carbon dioxide while reduce NAD to NADH, while alpha-ketoglutarate also will be oxidized to succinyl-CoA by alpha-ketoglutarate dehydrogenase, also release carbon dioxide and reduce NAD to NADH. Succinyl-CoA will be converted into succinate by succinyl-CoA synthase and produce GTP. Then, the succinate will be converted into fumarate by succinate dehydrogenase and produce QH2, which used to generate FADH2. The fumarate will be converted into mylate by fumarase, while the mylate also will be converted into oxaloacetate by mylate dehydrogenase and reduce NAD to NADH. All product of NADH and FADH2 from the Krebs cycle will be used into electron transport chain. Oxidative phosphorylation is combination of electron transfer chain and also chemiosmosis, which is the production of ATP from electrochemical gradient. Firstly, we need to understand the mitochondria structure. The outside one is outer mitochondria membrane, the inside one is inner mitochondria membrane, and the space between them is intermembrane space, and the one inside the mitochondria is a matrix. NADH and FADH2 from Krebs cycle will be used up in this electron transport chain. NADH will approach complex 1. Before that, it will be oxidized into NAD+. NADH lost an electron to the complex 1, and then the electron will be supercharged the complex 1. Complex 1 become more energetic and pump up more proton towards intermembrane space. This will create electrochemical gradient with higher concentration in intermembrane space rather than in the matrix. Electrons from complex 1 will be transferred to CoQ. Meanwhile, FADH2 will be approach complex 2, and then FADH2 undergo oxidation to form FAD. FADH2 loss of electron to complex 2. But complex 2 cannot be supercharged and cannot pump proton, so the electron from complex 2 will be transferred to CoQ together with complex 1 electron. Electron from CoQ will be transferred to complex 3. The mixings of electron from complex 1 and complex 2 supercharge the complex 3, create higher energy potential to pump up more protons to the intermembrane space. Electron from complex 3 will be transferred to complex 4 by more bicarrier cytochrome C. And then it supercharges the complex 4, which pump up more proton towards the intermembrane space. And while the electron being transferred to the last electron acceptor, which is oxygen, which splits into 2 oxygen ion and combine with proton to form 2 molecules of water. Now we move to the chemiosmosis, where is the production of massive amount of ATP. Before that, ADP need an energy input or catalyzed to form ATP. Therefore, here comes the ATP synthase. The higher concentration of the proton will be flow through the gradient into lower concentration, which is the matrix, to the ATP synthase. Then, ADP will make use of the proton to form ATP. As conclusion, all of these processes are important for living organisms such as plant and animal to carry out their metabolic activities. That's all from us. Thank you.